The overall goal of this procedure is to detect and quantify the DNA oxidation product 8-oxo-DG in biological samples by high-pressure liquid chromatography with electrochemical detection, or HPLC-ED. This is accomplished by isolation of a DNA sample from biological samples, followed by complete DNA digestion in a manner that minimizes undesirable DNA oxidation that can occur during sample preparation through the inclusion of metal chelator and a special DNA isolating reagent. Next, HPLC-ED run conditions are optimized, and standard curves for 8-oxo-DG and 2'-deoxyguanosine are constructed. The final step is to run digested DNA in series with pure 8-oxo-DG and 2'-deoxyguanosine in the HPLC in order to find the 8-oxo-DG to 2'-deoxyguanosine ratios in the digested DNA samples under investigation. Ultimately, 8-oxo-DG detection and quantification by HPLC-ED is used to quantify the extent of DNA oxidation in biological samples.
Yeah, the main advantage of this technique over existing methods is that it consolidates all relevant information from several published sources, in addition to providing rapid and straightforward means of testing the success of the deployment of this method in a new laboratory setting.
This method can help answer key questions in biomedical and toxicology research, such as how oxidative stress, or more specifically DNA oxidation, is mechanistically linked to adverse effects in cultured cells or experimental animals.
Generally, people new to this technique will struggle because there are many parameters.